everyone, so today in this video, me and my group members will be presenting about how microbes contribute to forensic science based on the paper we reviewed, Microbes, the Invisible Codes of Forensic Science. Established methods like DNA profiling and fingerprint analysis is the first thing comes into when identifying suspects, but there are limitations of these traditional methods. They struggle with sample degradation and cannot easily infer geographic origin, estimate the postmortem interval (PMI), or accurately reconstruct specific contact events. Microbiology has evolved from a niche discipline which is originally driven by biocrime, bioterrorism, and public health emergencies into an in-depth field integrated into mainstream forensic science. Thanks to modern advancements in massively parallel sequencing (MPS) technologies and machine learning algorithms such as random forest and deep learning, microbiome profiling has expanded from a minor complementary tool into a broader, highly relevant component of the modern research. Microbes are dynamic living systems. Unlike human DNA, which stays the same, microbial communities are alive and constantly reacting to their surroundings. This means they naturally record time and place. The relevancy during decomposition also plays an important role. During events like decomposition, the invisible tanato microbiome internal and epinecrotic microbiome external undergo highly predictable chronological successional changes driven by nutrient fluxes. These shifts transform the bacteria from passive non-relevant organisms into chronological witnesses capable of estimating the timeline of death, which basically means that when a body decomposes, the bacteria change in a highly predictable step-by-step timeline. Scientists use this timeline to calculate exactly when someone died. Microbes also adhere to the distance decay pattern. Their community similarity declines as geographic distance increases. This allows trace microbial evidence found on a suspect's belongings like shoes or car carpets to actively link them back to a specific crime scene location or burial site. Microbes change from place to place. If a suspect has unique soil bacteria on their shoes, it can link them directly to a specific burial site or crime scene. Microbes also have high individual specificity. Bacteria living on our skin are highly specific to us. Even when human DNA at the crime scene is too damaged, degraded, or low quality to use, the human microbiome can still be gathered and used to identify a person. Unless you undergo heavy medical treatments or major lifestyle shifts, your core personal microbiome stays mostly the same over time. Studies show that if you take a bacterial sample from a person today, it is unique enough to successfully match it back to them an entire year later.
Hi everyone, now about the time and place environmental applications. My focus is on the external application, which is determining exactly when a crime happened and where those involved has been. Let's start with the time by looking at what we call the clock of death. When an individual passes away, the strict physical barrier that keeps our internal organs shall collapse, and what was a highly regulated living ecosystem turns into the rapidly shifting, decomposing environments. This internal microbial community is known as the tanato microbiome. Because tissue breaks down into the highly specific chemical order, different microorganisms flourish and reduce in the predictable sequential wave. When a body is too degraded for the classical pathology, these microbial sections act as the incredibly resilient biological clocks. To read these clocks, we utilize advanced machine learning algorithms like random forest and deep neural networks. By feeding this model data on the changing microbial abundance, it can map out a precise timeline of death. In fact, recent research shows that the algorithm analyzing microbes on the head skin are exceptionally accurate, predicting the early to mid stage of death with the incredibly narrow margins of error of just over 3 days. But microbes don't just tell us when, they tell us where. When remains decompose outdoor, an immense burst of nutrient-rich fluid hit the earth, violently disrupting the native soil microbiome. It substrates baseline bacteria and sparks the massive bloom of opportunistic decomposition-driven microbes. What incredible is the durability of these footprints. These biological anomalies remain distinctly in the soil profile for up to 2 years even if the body has been entirely removed or relocated. By tracking this signature through the environmental DNA sequencing, forensic teams can easily locate distinct grave sites. This brings us to the trace evidence and geolocations. Every geographic area has a unique microbiome of place, shaped by the local soil, dust, water, and climates. When a suspect walks through the crime scene, their clothing and footwear forcibly trap these localized environmental microbes. Using statistical tools like a straws tracker model, forensic scientists can extract microbial DNA from the mud on the suspect's shoes and cross-reference it with the baseline spatial maps. The model then can calculate the exact statistical probability that the soil on their shoes match the unique microbial ecosystem of the crime scene. While human skin microbial profiles are remarkably stable and can persist on the physicals for up to weeks, we can have an account for the limitation like cross-contamination from the cohabitations. Ultimately, forensics microbiology, our perspective, is proof that the environmental itself is a silent, living witness that record timelines and movement with the microscopic precision. Thank you. Now I will pass it over to the presenter.
Basically, turning bacteria from a crime scene into forensic evidence involves four main steps. These invisible microorganisms can help forensic scientists identify where evidence came from and even estimate the time since death. Step 1. Microbiological sample collection and DNA extraction. First, bacteria are collected from surfaces such as door handles, soil, or object touched by the suspect using sterile swabs or scraping tools. In the lab, the bacterial cell walls are broken using physical and chemical lysis to release the DNA. Since only a small amount of DNA is usually present, a DNA extraction kit such as the Kiam DNA Investigator Kit is used to isolate microbial DNA while removing contaminants. Step 2. Next generation sequencing (NGS). The extracted DNA is analyzed using NGS, which can read millions of DNA fragments simultaneously. There are two main approaches. The first is cysteine as rRNA sequencing, which identifies the bacterial species by targeting a specific gene, similar to a barcode. The second is shotgun metagenomics, which resequences all DNA in the sample. This provides more detailed information about the bacteria, including their function and their interaction with the environment. Step 3 is bioinformatics. After sequencing, the result is raw DNA data. Bioinformatics software such as KIME2 or Motor filter out low-quality data, groups similar DNA sequences, and compares them with databases like Silva or NCBI. This produces a clear list of bacteria present in the sample. Step 4 is artificial intelligence and machine learning. Finally, the bacterial profile is analyzed using AI, such as a random forest model. Since the dataset contains thousands of data points, AI can quickly detect patterns that are difficult for humans to recognize. Based on these microbial signatures, it can estimate the exact time since death and the exact origin of the forensic.
In this section, I will discuss the limitations, challenges, and future outlook of microbiological forensics. Although microbiological forensics shows great potential in criminal investigations, several limitations still exist. One major challenge is sample collection and handling. Microbial communities are very sensitive to environmental conditions such as temperature, humidity, and contamination. If samples are collected improperly or stored too late, the microbial evidence may degrade and affect the reliability of the results. Another limitation is the overlap of microbial signatures between environments. Similar microbial communities can exist in different locations, such as public transport, homes, or workplaces. This makes it difficult to directly link a suspect to a crime scene with complete certainty. In addition, factors like age, gender, ethnicity, hygiene, and lifestyle can influence an individual's skin microbiome, causing variations in the microbial profile over time. There also exist legal and technical challenges. Since microbiological forensics is still a relatively new field, many courts do not yet fully recognize microbial evidence as strong legal proof. The field also requires advanced technologies such as next-generation sequencing, artificial intelligence, and machine learning, as well as experts trained in microbiology and forensic science. These requirements can increase the cost and complexity of investigations. Despite these challenges, the future outlook of microbiological forensics is very promising indeed. Researchers are currently working on combining microbial data with human DNA analysis to improve the accuracy of forensics identification. Artificial intelligence is also expected to speed up microbial analysis and help investigators reconstruct crime scenes more efficiently. Furthermore, the development of global microbial reference databases may allow scientists to compare microbial evidence more accurately across different regions and populations. Microbiological forensics may also play an important role in biosecurity and bioterrorism investigations in the future. So overall, while microbiological forensics still faces scientific and legal challenges, continuous research and technological advancement are helping it become a valuable tool in modern forensic investigation.
Some of microbial forensic analysis use material traces left at the crime scene to identify suspect, link evidence, and estimate the time since death or origin of evidence, especially when fingerprints or human DNA are not available. However, challenges such as data standardization, legal acceptance, and environmental factors still need to be addressed. Therefore, results must always be supported with reliable scientific database and careful interpretation. Hence, combining DNA sequencing and AI have great potential to make microbial forensic as a practical tool in a future criminal investigation. It is because sometimes the smallest microbial traces can provide the biggest clue.